Okay, so as I was saying, the only thing that you have to be careful about when you're doing these translations is that if you get to a stop codon, you do not translate past that. But apart from that, you just sort of carry on. These are pretty straightforward to do. The other version of these that you might see are the circle charts. And so for these, you start inside and you work your way out. So in this instance, if I start UAC, I go in the center, so I'm at U. I trace my way to A, and I trace my way to C, and that is again going to get me to a tyrosine. And if I do the next one, the GAA, I'm going to start in the G quadrant, go over to the A, and that's going to give me a glutamine. If we just do one more for good measure, CGC, I go to the C corner, go to G, go to C, that's going to give me the arginine. And so again, same rule for these, if you hit a stop codon, you stop translating. And so we did look at some steps of the different processes of how we would say replicate DNA. So as we know, DNA is double-stranded. So in order to get access to these strands, we need a special enzyme called helicase, which is this guy right here. And as you can see, helicase is like unwinding the DNA. So that other enzymes can get in here. So step number one, helicase unwinds DNA. Okay. And then step number two is you have a different enzyme, RNA primase, right, which is coming in here to add these little RNA primers because DNA polymerase, which is right here, cannot invent strands on its own. It needs RNA polymerase to do that. And so once those RNA primers are in place, then DNA polymerase right here can start adding nucleotides. So if we look, right, RNA primase, right, we'll be putting these primers on here. And then once I have one, DNA polymerase can then come in and start building that new DNA strand. So step number one, helicase unwinds the DNA. RNA primase comes in to add these RNA primers. And from there, DNA polymerase can then build a polymer by adding nucleotides to the 3' end, 1, 2, 3, for DNA replication. For mRNA transcription is a simpler process. You need only one enzyme, which is RNA polymerase. And so that does the job of unwinding the DNA. And it is also by itself adding those complementary nucleotides to build an RNA polymer. That's why it's called RNA polymerase. For translation, that's where you're going from mRNA to the polypeptide sequence. So just a reminder that translation depends on the ribosomes, right? And so you will have a messenger RNA. And what needs to happen first is that the ribosome itself needs to assemble on that start codon. So that's the initiation. And then at that point, tRNAs, right, with their anticodons and carrying some amino acid are going to start coming in and try to find matches, right? So step number one, ribosome assembles on the mRNA. They are initiated with the special AUG methionine tRNA. So that's the initiation. And then once it's going, more tRNAs are going to come in. When it finds a matching codon, that polypeptide is going to elongate. And then once it comes to a stop codon, the whole thing will let go. And so you've now completed your polypeptide. So that was all of 2.7. So in 2.8 was all about cell respiration, right? And so cell respiration is primarily the breakdown of glucose for some kind of ATP energy, right? And we had two options. Aerobic respiration, where oxygen is present, versus anaerobic respiration, where there is no oxygen present, right? And so we have this big whole like mind map thing. Whole point is to break down glucose to get energy in some kind of circumstance, right? So glucose during glycolysis is being broken down into pyruvate, and you get a small amount of ATP from there. And once I have that pyruvate, I can either go into the anaerobic respiration, where there is no oxygen, or the aerobic respiration that occurs in the mitochondria with oxygen. So ideally, I would always use the aerobic respiration because that's going to get me a huge amount of ATP, specifically 36 ATP. And then as waste products, I will also produce water and carbon dioxide with aerobic respiration. If I do anaerobic respiration, we looked at two different types of anaerobic respiration. The lactic acid fermentation, which occurs in animals, and as the name implies, produces lactic acid, or lactate. And so anaerobic respiration will always produce at least 2 ATP, and that is way less than the aerobics 36 that I would get. But lactic acid fermentation will also produce 2 lactic acid molecules. And we also looked at alcohol fermentation that occurs in yeast, right? And so this is what we use to make either yeast or bread. And so that will produce carbon dioxide, which it has in common with aerobic respiration, but it will also produce ethanol. So that is not something that is seen in any of the other types of respiration, only with the alcohol fermentation in yeast, but it will also have those 2 ATP. And so, make pasta eternal. That's like from an old to-do list. How weird that that is there. Anyway, so all types of respiration are going to overlap in that they all make ATP. That is for sure a shared product between all three. Aerobic respiration is unique in that it is only with oxygen. It is the only one that produces water as a waste product. When we're comparing aerobic respiration and fermentation in yeast, they both produce ATP and they also both produce carbon dioxide. But when I'm comparing, say, lactic acid in animals, it, like alcohol fermentation, is anaerobic, occurs without oxygen, but it is the only one to produce lactic acid. So that is unique. So be ready to do the compare and contrast, right, where you keep straight who's making what. And yes, yeast is the only one that makes alcohol that is unique only to that one. So we also looked at experiments in 2.8 related to the respirometer, and you are going to see these in paper 3 as well. And so just a reminder, the respirometer is following the consumption of oxygen. As respirating organisms, we do not create oxygen, we're not plants. So this is where you take some kind of creature and you're measuring the rate of oxygen consumption, like how much oxygen is this little guy breathing in. It requires some sort of alkali, some sort of basic solution to absorb the carbon dioxide. We all know that we breathe out, we're breathing out carbon dioxide. So if we want to follow only the consumption of oxygen, there needs to be some sort of alkali present to absorb that CO2. When the respirometer is working correctly and you're measuring consumption of oxygen, the oxygen is going to get pulled in, which is going to pull the liquid in behind it, right? And so that's how you can measure a rate of oxygen consumption with the respirometer. In 2.9, we talked about photosynthesis, right? And so we know that photosynthesis is the conversion of light energy into glucose somehow, and that we're using light to do that, that this is something that occurs in the chloroplast in plants. Some limiting factors of photosynthesis are the same as substrate concentration that we saw in enzymes, because plants, right, have enzymes just like we do, just like bacteria do. And so those enzymes are what is needed for photosynthesis, and those enzymes are going to be affected the same way as any enzyme would. So if you think about a plant, you know that plants need light. We know that they need carbon dioxide. So those are substrates of enzymes that are present in the photosynthetic process. So just like the regular substrate concentration, light intensity and carbon dioxide are going to be a limiting factor of photosynthesis. It needs it to do photosynthesis. And just like a regular enzyme, there's only so many enzymes, right, located in this chloroplast. So at some certain point, right, where I have low light, the enzyme doesn't have enough light to do its job, so it's not really making glucose. As I increase light, those enzymes now have a substrate to work on. They are going to build lots of glucose, but at some point, no matter how much more light I put on it, I have used up all my enzyme. And so this reaction is going to plateau. Same with carbon dioxide, right? If you're HL, you know Rubisco is the enzyme that's pulling carbon dioxide out of the air and fixing it into what it will become glucose. So there's a fixed amount of Rubisco. When I have only a little bit of carbon dioxide, my Rubisco is just sitting there. It's not able to build anything. But as I increase carbon dioxide, at some point I will have more carbon dioxide than enzyme, and it won't make the reaction go any faster because the enzyme is used up, right? So this is identical to the substrate concentration we saw in 2.5. And so again, plants are governed by enzymes, and so there's going to be an optimum temperature. When it's low, the substrate and enzyme aren't moving around very much, so you don't have a lot of product. There will be some optimum, after which, if you keep increasing temperature, those enzymes, which are proteins, are going to denature, right? So it's the same effect for photosynthesis as you would for any enzyme-driven process. And so photosynthesis, right? You're using light to build something, and so it's just a reminder that you have to know the nanometers of some of these wavelengths of light. That blue and red light are the most abundant, and so the pigments in plants are going to be primed to primarily absorb blue light, which is 450 nanometers-ish, and red light, which is somewhere between like 650, 600-ish nanometers. The reason that plants appear green is because they're reflecting green light. They don't absorb it. They're absorbing energy from blue on red light. Blue at 450 nanometers, red around 650. Reflect green, absorb red and blue. Just think like Jefferson, those are our school colors. Okay? So that fact accounts for both the action and the absorption spectra that we see. So this one is an absorption spectra. Absorption spectras measure the absorption at different wavelengths of light. It usually says right here, absorb, right? And so this is lining up the different pigments. It's shining different wavelengths of light on them, and it's saying how much light did you absorb at that particular wavelength. And so you usually see peaks around the 450 and around the 650, right? So absorbs in the blue, absorbs in the red. You don't get absorption around the green. It reflects it. That's why they look green. So these pigments can also be separated by the paper chromatography. Just remember, absorption spectra, different than action spectra. Absorption spectra measures absorption. Action spectra measures action. It's the rate of photosynthesis, right? So this is one way that we can track the rate of photosynthesis. And as expected, right, because these pigments are absorbing at 450 and around 650, you're going to see peaks around the same place, 450 nanometers and 650 nanometers. That red light and blue light, you get a nadir around green because it's not absorbing, it's reflecting at that. So the experiment that we looked at for photosynthesis had to do with paper chromatography, right? And so this is where you grind up a leaf in some sort of nonpolar solvent like alcohol or acetone. And so what you're doing, why it needs to be nonpolar, is because these pigments are embedded in the membrane of the chloroplast. And if you're HL, right, these guys are hiding out in photosystems 2 and 1. So they're in the membrane. And so if you want to dissolve them, you need to use a nonpolar substance because the membrane, the interior of the membrane is nonpolar. It's hydrophobic. So if you want these to move, you need to dissolve them in a nonpolar solvent. Yeah, because they're sitting within the membrane here. And so what you do is you draw the line on a strip of paper, you put the mush at that line, and then you dip that in some kind of solvent, and what's going to happen over time is that the pigments will separate. So we tried this, it didn't exactly work for us, but this is what's supposed to happen. So what this will allow you to do is to identify the different pigments by how far they traveled. And so you do that by calculating something called an RF, right? So the RF, right, what this is, RF is going to be the distance get back here, is the distance I can spell. Distance of this divided by the distance of that, right? So distance of the pigment divided by the distance of the solvent is going to give you that RF. And so you can use that number to identify your different pigments. Okay, so that is all of topic 2.